A specific section of DNA, called a gene, is converted into a polypeptide through a process known as polypeptide synthesis. A chain of amino acids is also known as a polypeptide. This is the building block for protein molecules, which are made up of one or more polypeptide chains, each of which has a particular purpose in the body. Transcription and translation are the two phases of polypeptide synthesis. The first step, transcription, takes place in the nucleus. DNA is a molecule made up of nucleotides with deoxyribose sugar, phosphate, and nitrogen bases, which all store the genetic material for organisms. These bases, which go by the name adenine, thymine, cytosine, and guanine, are joined by hydrogen bonds. A and T are joined by 2 hydrogen bonds, whereas C and G are joined by 3 hydrogen bonds. The DNA gene is unzipped during transcription by the enzyme RNA polymerase, which also dismantles the hydrogen bonds binding the DNA nitrogen bases together. On the DNA non-coding strand, it joins unbound RNA nucleotides to the matching DNA bases. The nucleotides of the RNA are identical, but uracil is used in place of thymine. Thus, an A on the non-coding strand now corresponds with U. RNA polymerase binds with the RNA nucleotides to form a molecule known as messenger RNA or mRNA, which includes a copy of DNA instructions for coding proteins. When mRNA splits from the non-coding strand, it exits the nucleus and travels to the cytoplasm, where RNA splicing removes the introns or non-coding sections of the RNA, leaving the exons or the coding regions of RNA. The second stage, translation, can now begin because the mRNA molecule is developed. This occurs in the ribosome. Ribosome is employed in translation to turn the mRNA code into a polypeptide. Ribosomes are ribosomal RNA-based organelles that are divided into a small and big subunit. In order to be decoded, mRNA binds to the small subunit and proceeds along the ribosomes. CGC is an example of a codon in the sequence that codes with the amino acid arginine. Codons are groups of 3 bases that are written by mRNA. Start codon, in this case AUG, codes for methionine, which is where mRNA signifies the start of the amino acid sequence. The molecule tRNA binds to the major subunit where each codon is read one at a time. The tRNA then takes free-floating amino acids that correspond with the mRNA bases in order to form a polypeptide chain. A developing polypeptide chain is created as amino acids are added by creating peptide bonds. The polypeptide chain ends with a stop codon UGA, as it is the end of the mRNA sequence. The polypeptide is released from the ribosome, where it then winds and folds into a distinct form due to the interactions between various amino acids. The function of this shape is then determined. However, DNA mutations might happen which have an impact on the final polypeptide chain generated. For instance, a single base pair in the DNA sequence can be changed, added, or removed to cause a point mutation. In this non-coding strand, a point mutation in the DNA sequence results in the substitution of an A for a C. Due to the U being replaced by a G during transcription, the mRNA sequence is slightly different from the original. The ribosome reads the mRNA codon once the mRNA enters from the cytoplasm for translation, and the tRNA transports the relevant amino acids. Following a point mutation, the original codon UUC, which codes for phenylalanine, has been altered to GUC, which codes for valine. This framework leads to a marginally altered amino acid sequence, which will have an impact on the interactions between amino acids and the polypeptide's overall shape. This shape modification will impact the way the protein behaves within the body. A nitrogen base in the beta-globin chain, which codes for the protein hemoglobin, or sickle cell anemia, is an example of a genetic disease brought on by substitution point mutation. In transcription, a nucleotide that would normally be a T in the non-coding strand is changed to an A, thus changing the mRNA codon from GAA to GUA. Instead of the hydrophilic amino acid glutamic, the altered codon GUA codes for the hydrophobic amino acid valine during translation. The distinct characteristics of the amino acid alter the structure and function of hemoglobin. This is the protein that carries oxygen within red blood cells. This then causes red blood cells to change from being round and flexible to being stiff and sickle-shaped, which makes it difficult for them to pass through blood channels. These affected organisms are at high risk of blood clots and ineffective oxygen transfer. This mutation affects an organism negatively, even fatally, and will prevent the continuity of the species.